And that's how the Honorato project was born, and the creators of this project are the members of the team iGEM USP-EEL. Great question. The name is a tribute to Brazilian folklore, with Honorato being an indigenous person in the form of a giant but kind snake who has his curse broken by a brave soldier. Tropical regions have exciting myths and legends and a vast culture, but they also have a problem: neglected tropical diseases. And according to the World Health Organization, WHO, snake bites are one of them. According to WHO, it is estimated that annually the number of people bitten by snakes can reach 5.4 million, and of that amount, the number of deaths varies between 81 and 138,000. In addition, up to 400,000 victims are incapacitated due to the action of the toxin in the bloodstream. In Brazil, most accidents with snakes are caused by snakes of the Bothrops genus. According to data from 2019, there were 21,009 cases of bites from snakes of this genus. 10,687 of them were considered mild cases, 7,971 were considered moderate cases, 1,468 were considered severe cases, and 883 cases had no record of evolution. More than 238,000 were also notified, and there was no identification of the type of snake, showing that the number of bothropic accidents could be even higher. Snake venom is 90 to 95% protein. Phospholipase A2, or PLA2, are the main component enzymes of the venom, being also the most harmful to the affected cells. They are mainly responsible for muscle necrosis, also known as myonecrosis, which can cause loss of limbs, in addition to shock and death. But snakes have their own inhibitors for PLA2. Because of this, a snake cannot die infected by its own venom. These inhibitors are called phospholipase inhibitors, PLI. Based on amino acid sequence homology, PLIs are divided into three classes: alpha PLI, beta PLI, and gamma PLI. Our solution is to produce gamma PLI via synthetic biology, as it is able to inhibit most classes of PLA2, while other classes of inhibitors cannot. The objective is to offer a solution that can be used in emergency situations, in environments such as ambulances and emergency rooms, even before the identification of the species of snake. We also hope that our project can contribute to a research base that aims to study the fall of fatal cases and loss of limbs due to snake accidents, providing a protocol on the production of recombinant gamma PLI and tests of its effectiveness for the inhibition of PLA2. Currently, the anti-venom serum is produced through the wide use of large animals, such as horses. In this process, the snake's venom is injected into the animal in successive doses, and after days with the venom in its body, the horse's blood is collected with the presence of antibodies capable of fighting the venom. In addition to the damage caused to this animal during the process, the conventional production of anti-venom serum has less control and greater difficulty in purification when compared to the proposed process via synthetic biology. Due to the COVID-19 pandemic, it was not possible for us to perform experiments in the wet lab, so we focused our efforts on the dry lab. Well, how were our experiments and what were the results? The modeling of the project started with the following question: how can we know how efficiently our recombinant protein inhibits the venom? To answer that question, we did a lot of research to determine the kinetics and structure of the protein in question. First, to discover information concerning kinetics, we contacted the researchers and authors of scientific articles related to PLI. We've been told that the PLI plus PLA2 system appears to follow a competitive inhibition. Assuming Michael's kinetics, we obtain an equation that describes the substrate concentration as a function of time. In cellular medium, after the inoculation of the venom, the substrate is the phospholipids of our cells. However, to analyze the consumption of the substrate in the lab, we use a compound called 4-nitro-3-octanoiloxybenzoic acid, which has similar molecular structures to compounds present in the membrane of affected cells. The acid reacts with the enzyme PLA2 and does not react when inhibited by PLI. The kinetics will be studied with a spectrophotometer, as the substrate can have its concentration read according to the absorbance. Regarding structural modeling, we discovered possible molecular structures of PLI using homology with similar proteins, as we know that this enzyme has a high similarity between snakes of the same genus. Using different proteins, we use a software to elucidate several possible structures of PLI, which can help future researches. So, we were able to predict the behavior of enzyme kinetics with different concentrations of PLI, making it possible to obtain the ideal concentration of this enzyme in the inhibitor used. Furthermore, structural modeling will be useful in future studies of the enzyme, and the combination of our results can be an initial steps towards the 100% synthetic production of an anti-venom serum. But what about beyond the dry lab? Our objective beyond the laboratory was to talk to the population about the implication of snakebite accidents, first aid, how the snakebite serum is currently made, and the importance of preserving snakes. In order to act in the best possible way, we contacted the population as well as professionals working in the health area such as researchers, nurses, and doctors. As a conclusion of our research, we observed two main points that we could work on: the misinformation on how to act in case of snakebite accidents, and the stigma on genetically modified organisms. For both issues, we seek to dialogue with the population through social medias using audiovisual resources. In order to do so, our first aid project and measures were published in the University of São Paulo newspaper in written form, on TV Metropolitana communication channel in the state of São Paulo through an interview, and we also participated in the Mammoths of Science podcast, which is a podcast focused on scientific dissemination. The University of São Paulo has a technical high school in the city of Lorena, so we saw an opportunity to disseminate knowledge in synthetic biology and information about snake accidents. Thinking about the students' curriculum and their objectives for the college entrance exam, we set objectives that encourage their studies beyond biology, such as writing an essay in the standard format of the National High School Examination, also known as ENEM, which is the largest form in Brazil for admission to universities. Also, by talking to professionals and researching Brazilian legislation, we better understood how the drug approval process works and its implications. In the end, we learned way more beside the fact that it is very difficult for a snake to die with its own venom.